Good morning and good afternoon, everyone. Welcome to today's webinar entitled The Growing Concern of Off-Target Effects, How to Monitor and Minimize Off-Targets in your Genome Editing Experiment. My name is Louise Baskin. I'm going to be the moderator for today's event. This educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. This presentation is educational and offers continuing education credits. So just click on the continuing education window at the bottom of your screen to obtain those credits. And we do encourage you to participate today by submitting any questions you might have during the presentation. To do this, you just type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. And you can also submit any technical issues using this method as well if you have any trouble seeing or hearing the presentation. But with that, I'm going to turn it over to our speaker, Pandhita Pochanard. She is a product manager for genome editing at Thermo Fisher Scientific. So Pandhita, I'll turn it over to you to begin your presentation.
Hi everyone, my name is Pandhita Pochanard and I'm the product manager for genome editing at Thermo Fisher Scientific. For today's presentation, I'll be talking in detail about the growing concerns of off-target effects in genome editing experiments. I will discuss available methods and strategies that can be taken to monitor and minimize off-target effects for your application. Just wanted to go over quickly the agenda for today. We'll start off with talking a little bit about the CRISPR Cas9 system, then go into a little bit more detail about what off-target events are and why you should be concerned, and follow closely with strategies to reduce off-target events. And finally, talking about the Invitrogen TrueCut HiFi Cas9 protein, and then close off the discussion with a summary and Q&A. Genome editing has become exceedingly popular in common places due to the CRISPR-Cas9 revolution. CRISPR-Cas9 has been a significant paradigm shift in functional genomics research and has made genome editing easier and more accessible to a wide array of researchers like never before. I would like to start off with a quick overview of the CRISPR-Cas9 system. CRISPR-Cas9 is an RNA-guided nuclease system that was first identified as an adaptive bacterial immune response pathway. The specificity of the system is due to the RNA-guided nature of the system. It comprises of two main components, the guide RNA, which contains the sequence that guides the protein to the target location. It is a duplex of CRISPR RNA and tracer RNA molecules. And then the second, equally as important component of this CRISPR-Cas9 system is our Cas9 nuclease. It's a protein that performs the actual cutting or cleavage once guided to the intended location by the guide RNA. The CRISPR-Cas9 system is dependent on what we call a protospacer adjacent motif, or PAM site, for the guide RNA to recognize the target site on the genome. The CRISPR-Cas9 system can be delivered into the cells in three different formats: plasmid, RNA, or protein. Many researchers actually favor the delivery of the protein RNA complex, or RNP, which we'll talk in a lot more detail later in this presentation. So first, I want to introduce one of the highly successful genome editing products developed by Thermo Fisher, the Invitrogen TrueCut Cas9 Protein V2. It is the wild-type Cas9 protein that's been engineered to give it a very high specificity into the nucleus of the eukaryotic cells. This allows it to consistently deliver high editing efficiency in all tested cell types. We also have several validated protocols in numerous cell types available to help researchers get the highest efficiency and success with our protein. And as you see here on the slide, some of the data in the cancer cell line on the right-hand side. We also demonstrated high editing efficiency when using TrueCut Cas9 Protein V2 in more therapeutically relevant cell types such as primary T cells. Here on the slide, you see on-target editing efficiency approaching 100%. For more information on the data as well as the protocol, please visit thermofisher.com where an application note is available on this particular experiment. With CRISPR-Cas9 holding so much promise and with increased adoption, there are emerging concerns about off-target events in CRISPR-Cas9 mediated genome editing. Now, off-target events occur when unintended edits happen, so this could potentially add another variables or unknowns to your experiment. This is particularly important to monitor and minimize off-target effects, especially for therapeutics application, to make sure that any risk associated with these undesired edits are controlled or reduced for your experiment. As you can imagine, with off-target events, you don't know where the edits may have occurred. As such, depending on where the edits happen in the genome, it could potentially affect the phenotypes of your cell models. As I mentioned previously, off-target effects can affect your genome editing experiment. One of the main reasons that researchers are concerned about off-target is when they are working towards products such for cell and gene therapy. In something as high value and well-regulated as these applications, it is important to use a system that will minimize the risk of undesired genomic changes. As a result, it is particularly important for therapeutics application that you are able to firstly screen or monitor for off-target effects, and in combination with that, implement strategies to minimize off-target effects in your experiment altogether. Some of the key strategies that we'll be discussing in more detail today include the use of RNP delivery format, high-scoring guide RNAs, or high-fidelity Cas9 to help reduce the overall occurrence of off-target effects in your experiment. So as I've alluded to previously, it is important to be aware of strategies that are available for you to implement to help reduce off-target effects in your experiment. So first off, on the left-hand side here, RNP delivery format. I'm not going to go into a lot of detail on this first strategy today, except to say that RNP, or protein guide RNA complex, is a favored format for its ability to provide an initial high level of CRISPR-Cas9 when introduced into the cells, followed by a rapid decay. And the reason for this is the higher or more sustained level of this CRISPR-Cas9 tool in cells typically lead to more off-target effects. So with the introduction of this tool as an RNP format and its ability to quickly clear the cells, leading to a reduced occurrence of off-target effects. Second strategy here is in using high-scoring guide RNA, and we'll spend a little bit more time on the next two strategies today. So firstly, with the use of high-scoring guide RNA, you can leverage an in silico design tool, such as our Invitrogen TrueDesign genome editor, to predict potential off-target sites for the intended edit you're looking to achieve. From this tool, you'll be provided with a ranked list of guide RNA based on score, and the score takes into account different parameters such as predicted efficiency, specificity, and proximity to the edit site. Now, one thing to note as it relates to high-scoring guide RNA is there are certain limitations that may prevent users from having a good variety or good choice of guide RNAs. This includes perhaps there may not be any or no PAM sites available near your intended target. As well, with the guide RNA, selection that is provided by the tool, the efficiency and or the off-target profile may not be optimal. And then the third strategy here is the use of high-fidelity Cas9. This is a Cas9 enzyme that's been engineered to demonstrate high specificity. What that means is it exhibits improved off-target profile by reducing off-target events. But then one of the trade-offs that we would have to keep in mind as it relates to all high-fidelity Cas9 is you would see some reduction in on-target editing efficiency when compared to our wild-type Cas9 protein. So just some of the things to keep in mind as we go through this presentation to talk in a little bit more detail on the strategies to reduce off-target events. So now let's dive into a little bit more detail on the role of guide RNAs and off-target effects. One thing I wanted to start off with is to say that the precision of the CRISPR-Cas9 system is driven by its ability to find the perfect match between the guide RNA and the target DNA. So when this happens, or when there is a perfect match, you get what's called a perfect duplex, or an on-target guide RNA design, shown here on the left-hand side. As you can see, there's no mismatches anywhere, and you just are able to achieve that perfect duplex between the guide and the target DNA. However, the system itself can actually tolerate some base pair mismatches, which can potentially lead to off-target effects or sites, shown here on the right-hand side. Off-target effects occur when there are base pair mismatches between the guide RNA and the target DNA, which means that the protein or the Cas9 didn't necessarily cut at precisely where we want it to cut. So again, when these off-target events happen, it could lead to unintended changes or edit within your cell model, which could affect the phenotypes of your model, right? So depending on where the off-target events occur, it could lead to unexpected changes because you don't know if those off-target events hit a vital coding gene. Is that going to kill my cells? You know, are the edits or the phenotypes that I'm seeing, is it because of my intended target, or is it because of these off-target effects? So again, going to the point of off-target effects just introduce another variable or unknowns into your model. To facilitate guide RNA design, we do offer the free online TrueDesign genome editor software, which is available here. You can visit our software at thermofisher.com/TrueDesign. The tool itself provides a fast and easy design of CRISPR guide RNA for your genome editing projects. So for all of the guide RNA designs, the tool will carry out an analysis of predicted off-target cut sites so that you can fully assess the predicted potential of each guide RNA specificity. Additionally, the tool also offers a one-stop shop environment where you can easily order other genome editing reagents, including Cas9, primers, and or transfection reagents in one convenient location. If you want to learn more about our TrueDesign genome editor, we have several tutorial videos, workflow guides, as well as a webinar available online so that you can, check it out if you would like to learn more about our software. Now, going back to the strategy, that second strategy around the use of high-scoring guide RNA to minimize off-target effects. Let's dive into a little bit more detail here. So what I'm showing is a screenshot from the Invitrogen TrueDesign software. It's showing the location of a desired knock-in of a fluorescent reporter. You can see here in the snippet up top. And then when the software scanned the nearby region, you can see that in the result table here that I'm showing that there are plenty of locus where there's a lot of adjacent PAM sites to choose from. You can see the column that says PAM site, right? And the algorithm scored, I'm showing here 5 different guide RNAs. So you can see that the algorithm scored the guides very highly, all of them above 98%, and each of them have no more than 2 predicted off-target sites, as shown here in the table. One thing to note, as I mentioned, by leveraging the software, you're able to quickly design or identify the guide RNA that's appropriate for your intended edit. And through this table, you have the option to pick the highest scoring guide RNA for your intended edit. One thing to note is for the predicted off-target sites, as I mentioned, that each of them have at least, at most 2 off-target sites. But for the predicted off-target for each guide RNA, you can also click on the link to get more information, as I'm showing here in my third image, more information on the predicted off-target, including location, as well as identity of the base pair mismatches. But overall, you can see that for this particular target location, there are actually plenty of high-scoring guide RNAs to choose from that has minimal off-target events. So in this situation, for this particular edit, you have a lot of options from a guide RNA design perspective to help minimize off-target events. So why not just always choose very specific guide RNA or very high-scoring guide RNA for all of your genome editing experiments? So the problem here is that researchers may sometimes be forced to use guide RNA designs with higher than desired off-target events. So in this example here, I'm showing, we're attempting an edit at a different locus this time. We're attempting to do a SNP, shown here in blue. And as you can see, for this particular edit that I'm trying to do, I only get 1 predicted guide RNA, and is only predicted to have a very moderate performance of about 74% with 35 potential off-target sites. So as a result, since I only have 1 guide RNA to pick from, this limitation, again, is like I don't have a lot of option or choice to work with, right? And one thing to note is generally around the limitation of guide RNA choices, it's mostly driven by, what the edit that you're trying to achieve, as well as the surrounding sequence space. There may be very few or no PAM sites available adjacent to the desired edit site. And in this situation, you may have to work with what you have from a guide RNA perspective. Again, you only have 1 guide RNA, and the scoring is not great. It's not very high, right, at 74%. And as you can see, with the number of off-target sites that are predicted of 35, this may not necessarily be the most specific guide RNA for you to work with. So in that situation, what are you going to do, since you're now limited from a guide RNA aspect? And the key takeaway here is not all edit sites have high scoring and specific guide RNA to choose from, right? As we've mentioned, choosing or using high scoring guide RNA is one of the strategies to help minimize off-target effects. So then I want to segue into our third strategy, which is around high-fidelity Cas9. So I first wanted to start off by talking about, what is a high-fidelity Cas9? A high-fidelity Cas9 is a Cas9 variant that contain additional point mutations. So it's been engineered with that additional point mutation to demonstrate improved specificity, or equivalent to improve off-target profile. So how does it work? So with these point mutation that's been introduced into the Cas9 enzyme, what it did is it destabilized the interaction between the Cas9 protein and the target DNA. So when this occurs, that's why for the HiFi Cas9 protein, it's less likely because of that disruption in the interaction between the Cas9 protein and the target DNA. What it does is the protein itself is less likely to bind to any off-target sequence due to the point mutation. So what this results in is, improved specificity or, improved off-target profile. But then on the flip side, you also lose a little bit on an on-target editing efficiency front. Also on the slide, you'll find a table comparing the performance of a wild-type Cas9 to a high-fidelity Cas9. So with our wild-type Cas9, we know, as I've shown you previously with our TrueCut Cas9 Protein V2, that it demonstrates excellent or really high on-target editing efficiency. But what it doesn't do so well of is that ability to demonstrate or reduce off-target events. Then on the flip side, with the high-fidelity Cas9 and the engineering that went into destabilizing that interaction, what you gain with the high-fidelity Cas9 is in its ability to greatly or significantly reduce off-target effects. And then, as I mentioned, you lose a little bit on the on-target editing efficiency when compared to the wild-type Cas9. So that's fundamentally the trade-off or the difference between wild-type Cas9 and a high-fidelity Cas9, right? So with that in mind, we set out to identify the best high-fidelity Cas9 protein candidate that would offer the superior off-target profile while still maintaining or preserving the high on-target editing efficiency as much as possible. So we've taken the following approach captured here on the slide in our quest to identify the best HiFi Cas9 candidate. So first, what we did was we screened several HiFi Cas9 candidates, both from an in-house candidates as well as those in published literature. We screen it for its off-target profile using next-generation sequencing. We then validate those off-target results using ion AmpliSeqtechnology. And then lastly, because we're trying to find that right balance between off-target and retaining on-target editing efficiency, we also evaluated the high-fidelity Cas9 candidates for its on-target editing efficiency as well. So again, we're really looking to find the best candidate that would offer the superior off-target profile that we expect from high-fidelity Cas9 while still maintaining the high on-target editing efficiency. So now let's talk a little bit more about the analysis tool that we use to screen for off-target events. So there are several detection tools available, including the in-cellular detection tools such as Guide-Seq that is very commonly used for off-target detection. And the idea with in-cellular method is it more closely represents the cellular nuclease-based gene editing environment as opposed to other in vitro detection methods. However, we know that for most of the available in-cellular tools, there are some issues related to the sensitivity, especially with its ability to detect low-frequency off-target effects. And this is primarily due to the non-specific amplification step of Guide-Seq. So to address this sensitivity issue, we leverage TEG-Seq, which is an adaptation of Guide-Seq that would offer a more targeted genome editing off-target screening method. And I'll go into that in more detail in my next slide. Here is a quick overview of TEG-Seq, which once again is an adaptation of Guide-Seq. It includes the introduction of a short double-stranded tag that is delivered into the cells along with the CRISPR Cas9 tool. This DS tag is integrated into the genome where the cleavage occurred. Then through the use of a phosphorylated primers, we're able to achieve more specific amplification, resulting in an output that looks very similar to the table shown here on the right-hand side. For more information on TEG-Seq, I've included here on this slide some publications that go into more detail about the method and procedure. I also want to note that through our custom biology services team, TEG-Seq is also offered as a custom service. So now that we've learned a little bit more about TEG-Seq, let's go into the first step that we took to identify the best high-fidelity Cas9 candidate. So here we've screened 7 different high-fidelity Cas9 candidates and benchmarked that against the wild-type Cas9 protein using TEG-Seq. On the left-hand side, you'll find the output from TEG-Seq, where the green row on the top represents the on-target location, and every other row beneath it represents off-target locations. So you'll find that as you look from left to right in the table, going from wild type all the way through the different variants of the high-fidelity Cas9, you see more zeros, which represents less off-target sites that were registered with TEG-Seq. Then on the right-hand side in the bar graph, here's a summary of the results. So as I mentioned, wild-type Cas9, we identified 34 off-target locations, and as we go through the different high-fidelity Cas9 variants, you see that drop in the number of off-target sites. With high-fidelity Cas9 variant 4, the orange bar on the most right of the graph, you see that with high-fidelity variant 4, it eliminated 75% of the off-target events that were detected when compared to wild-type Cas9. So again, just by going to the high-fidelity Cas9, we saw the best result with the HiFi variant 4, being able to eliminate 75% of off-target events when compared to wild-type Cas9. Then what we did was taking that TEG-Seq result, we now validating it using ion AmpliSeqtechnology. So on the left-hand side, you'll see a heat map. Again, the target here is the HEC4 loci. We did a similar comparison where the wild-type Cas9 is on the leftmost column, and then gradually go through the different HiFi Cas9 variants. So you'll see that the number in each of in the table represents off-target editing percentage. So once again, with variant 4, we saw that it has the lowest percentage of off-target edits. And then once we do an expanded, we screen across an expanded panel, this time looking across 24 different loci, we identified high-fidelity variant 4 on the right-hand side, the orange bar, as being the most specific. Again, this data right here is normalized against the wild-type Cas9. So you see that with the high-fidelity Cas9 variant 4, it only registered 39% off-target edits when compared to the wild-type Cas9. And then finally, once we saw that great off-target profile, as I mentioned, we want to check to make sure that with this high-fidelity Cas9 variant 4, which has the best off-target profile, that it's able to maintain also a high on-target editing efficiency. So what you see on in this slide right here, the bar graph on the left-hand side is once again the average percentage on-target editing normalized to wild-type Cas9. So you'll see the wild-type Cas9 having 100%, and we saw that with the other high-fidelity variant 1 and 2, it also was able to preserve a pretty high percentage of on-target editing, with the high-fidelity variant 4 exhibiting over 86% on-target editing efficiency, right? So it's able to preserve over 86% of what we would see with the wild-type Cas9. So that performance on the on-target editing efficiency front, together with the superior off-target profile and results that we saw in the previous slides, we then proceeded to select the high-fidelity Cas9 variant 4 as our new product, the Invitrogen TrueCut HiFi Cas9 protein. So in introducing our TrueCut HiFi Cas9 protein, it's a Cas9 variant that's been engineered to demonstrate a superior off-target profile while maintaining high on-target editing efficiency. Our data showed near complete elimination of off-target events at key loci in a number of cell types, including primary, immune, and stem cells. We saw better off-target profile with the TrueCut HiFi Cas9 protein when compared to the wild-type TrueCut Cas9 protein V2. What we also saw was a better off-target profile relative to other suppliers' HiFi Cas9. And then lastly, we were able to maintain that maximum on-target cleavage due to the unique Cas9 modifications and engineering that we did to our TrueCut Cas9 products. So over the next few slides, we're going to be going over some of the data using the TrueCut HiFi Cas9 protein in therapeutically relevant cell types. So we perform an experiment in human primary T cells as well as human-induced pluripotent stem cells. So what I'm showing first here is the data in primary T cells. So in using the TrueCut HiFi Cas9 protein, we saw fewer off-target events when compared to the wild-type Cas9 as well as a different supplier, HiFi Cas9. So what we did for this experiment is we used 21 guide RNA targeting 4 CAR-T-related therapeutic genes, including TRAC, CD52, TRAC-B, PD-1. And then, as I mentioned, we edited the cells using the TrueCut Cas9 protein V2, or our wild type, another supplier's high-fidelity Cas9, and finally benchmark that against our TrueCut HiFi Cas9 protein. So the dots on the slide, the red dots represent on-target events. So you see that in this graphic right here, the data is represented as the read per million, or RPM, at the off-target location over the on-target location RPM. So it's displayed as a ratio. The red dots represent on-target events, and each of the gray dots represents an observed off-target event. So you see that each of the enzyme, the result is divided into 4 different quadrants. Each of the quadrant represents the therapeutic genes that we've selected. So in the red box here, you'll find the results of the TrueCut HiFi Cas9. You see, if we look at PD-1, which happens to have the most gray dots across the different nucleases that we tested, that with the TrueCut HiFi Cas9, you see significantly less gray dots, which once again represent off-target event when compared to the supplier high-fidelity Cas9, as well as the wild-type TrueCut Cas9 protein V2. And then in general, if you look at the other 3 genes that we targeted, you also see significantly less off-target events when using our TrueCut HiFi Cas9 compared to the other 2 enzymes that we benchmark against. Right, so this further confirmed that by using our TrueCut HiFi Cas9 protein, we were able to significantly reduce off-target events across all target loci in primary T cells. Then we look, again, still in T cells, now we look at the on-target editing efficiency. In the bar graph shown on this slide, you see the average normalized % of on-target editing efficiency. We have split it between indels and HDR, but you'll see that with this data that's been normalized to the wild-type Cas9, we see that with the TrueCut HiFi Cas9 protein, we saw a higher editing efficiency when compared to the other supplier, HiFi Cas9. With the TrueCut HiFi Cas9 protein, we were able to preserve 84% in indel and 88% of homology-directed repairs, or HDR, when compared to wild-type Cas9. So again, really demonstrating that with our TrueCut HiFi Cas9 protein, from an on-target editing efficiency perspective, it performed nearly as well as the wild-type Cas9, again, being able to preserve over 80% for on-target editing in primary T cells. So on this slide, you'll find the TrueCut HiFi Cas9 protein performance in human iPSCs. So for this particular experiment, we picked 3 different genes to target: HBB1, HBB2, and HEC4. And the reason why we picked HEC4 is because it's a well-studied site for off-target effects in different cells. On the left-hand side, shown here is the dot plot graph, which, similar to what we saw in primary T cells, the TrueCut HiFi Cas9 registered significantly less off-target events, which is represented by the gray dots, as compared to the other 2 Cas9 enzymes that we benchmark against. And then on the right-hand side with the bar graph, this time we're showing the data specifically for HEC4. So in looking at the TrueCut Cas9 protein V2, or our wild type, we saw 9 off-target events registered. And then in looking at the other supplier, HiFi Cas9, we saw 6 off-target events. And finally, when using the TrueCut HiFi Cas9, we registered 4 off-target events. So that reduction that we saw from 9 to 4 when compared to the wild-type Cas9 protein represents about 55% elimination of off-target events when compared to wild-type Cas9. Right, so this further confirm and demonstrate again that by using the TrueCut HiFi Cas9, we were able to achieve that higher specificity in human iPSCs compared to both the wild-type Cas9 and the other supplier, HiFi Cas9. So in summary, what we learned from today's presentation is that off-target effects or cleavage is a problem for many CRISPR-Cas9 applications. Off-target events can lead to unintended edits, which could introduce a variable or an unknown into your genome editing experiments. We learned that there are several strategies that can be implemented to help minimize off-target effects. These include the use of RNP delivery format, the use of optimized or high-scoring guide RNA, and finally, the use of a high-fidelity Cas9 nuclease. And then we saw today that we were able to identify the optimal high-fidelity Cas9, which ultimately became the Invitrogen TrueCut HiFi Cas9 protein, which have demonstrated superior performance in reducing off-target events while still maintaining high on-target editing efficiency in both primary T cells and human iPSCs. So with that, I would like to thank you for your time, and we'll open to questions.
That was great. Thank you so much, Pandhita. We had a lot of questions come in, so we're going to go ahead and get to the question and answer session right away. If there still is a question you'd like to ask, you can still do so now. Just click on the Ask a Question box located on the far left of your screen. Okay, Pandhita, let's go ahead and get started. So the first question is, if my guide RNA has predicted off targets, how do I figure out if I actually have cleavage at those sites?
Yeah, thanks, Louise. That's a really good question. So for a lot of the experiments, right, like this sort of verification as far as checking if the off targets actually occurred isn't really necessary. Typically, that sort of verification is done when you're at that high value, so clonal cell line step. So essentially, the thought here is that by using a high fidelity Cas9 to reduce that off-target effects, you would have that peace of mind knowing that it's already going to be reduced. So it's not necessary that you would have to conduct a TEG-Seq analysis always just to check that the off-target effect happens. So that's sort of like the approach that we would recommend.
So it's really more peace of mind until you get to potentially a really high value clonal cell line or something where you need to verify.
Yeah.
Okay, that makes sense. Similar, obviously, question about off targets. There were a lot of off targets in the experiments you showed with the wild type Cas9. Were those guide RNAs predicted to be so nonspecific?
Yeah, again, another great question. It's good for catching that, right? So a lot of the cases in the data that we showed, we intentionally actually chose what we call a malicious or bad guide RNA, just because, just to see that we know that at this target, there's going to be a lot of off targets, so it's just more measurable and sort of meaningful to see that, to be able to see the reduction in off target effects when using the HiFi. And again,
right.
just that's sort of what it is. So yeah, that's exactly what we did. We intentionally picked the bad guide RNA to demonstrate the ability to reduce.
That makes sense. Yeah, otherwise there's not much to measure.
Right. Otherwise you're seeing like one dot or something, so that's not.
Right. So this question came in when you were showing the screenshots of the TrueDesign software. And so the question has to do with the score that's shown for the guide RNAs. And it asks, what does your score represent? Is that the predicted cutting efficiency?
Yeah, so again, great question. So for the score that we, in the screenshot that we showed, there's a column that says score. So that's actually so that scoring is not representative of the efficiency percentage, but what it does is it actually accounts for three different parameters, actually. So the first one being, efficiency is one of the parameters or factors that we take into account, the number of predicted off target, and also the distance to the target site. So it's a combination, it takes into account three different factors, and then it kind of gives you a score. And Louise, if do you have any additional information
to
add to that?
I think you explained that just right. I mean, it's exactly that, right? So the higher the score, the higher the predicted success, right? Not necessarily literally. If you see a score of 90, we're not saying that will get 90% cutting,
right?
But we're saying it's probably better than one that scores, 60 or 70%.
Right.
Exactly. Here's a nice straightforward one. Do you sell the HiFi Cas9 as a plasmid?
Good question. So no, we don't sell HiFi as a plasmid. We sell it as, RNP, right? Or as protein. So that's a good straightforward question.
Yep. And you made the point about the plasmid format being less desirable from a specificity standpoint in general, right? So you're not continuing to potentially, leave a nuclease in your cells longer than it needs to be to make its cut.
Yeah. Yep. That's right.
Okay, so next question, back to the TrueDesign. What is the difference between the guide RNA from the TrueDesign software and the ones available on your website?
Yeah, so I'm actually going to pass this one to you, Louise.
Sure, yeah. Kind of helps to have a second product manager on the line. So the guide RNAs that you find on our website, not in the software, those are pre-designed with an algorithm to predict highly functional knockout. So if you're just trying to knock out your gene, these guides go through, again, it's an algorithm for both efficiency as well as having a very rigorous specificity check. So for on the web, you can just search for your gene. We have pre-designs for human or mouse, and then you can just click to order. Whereas the TrueDesign software is really more focused as a tool for precise gene editing. If you're doing HDR, if you're doing knock-in, If you need to make a cut in a very specific region, that's really what it's designed for. And it also supports additional species beyond human and mouse. So if you're doing something really customized, we would direct you to the TrueDesign software tool. Thanks. Appreciate the chance to answer that.
Good answer, Louise.
Yeah. So back to your data, and this is kind of related to the specificity question about the guides that we used. What percent decrease in off targets should I expect to see?
Mm-hmm. So with the TrueCut HiFi Cas9, we tested it, as I mentioned in the presentation, in several cell types, primary T cells, iPSCs. So overall, in general, we are expecting at least 80% reduction. Again, that's going to depend on, the different cell types and sort of the intended target as well. But from the data that we generated, the benchmarking data, we do expect to see over 80% reduction in off targets compared to the wild type Cas9 version.
And I would think it's also really going to depend, kind of based on the data you showed. If you have a pretty non-specific guide with like 60 off targets, you might be able to reduce that, to 5.
Yeah.
But if you have a guide that only has 5 to begin with, maybe you only drop to 1 or 2. So the percentage is kind of going to be indicative, right, of the starting point. Kind of your earlier point about how good is the guide.
Right. Yeah, that's a good point.
Yeah. This is a good one. Will the TrueDesign software suggest when the HiFi Cas9 is necessary?
Yes, great question again. And as I mentioned, with the TrueDesign software and it being almost like a one-stop shop where you can order all your other genome editing reagents as well. But both versions of the Cas9 protein will be available for purchase through the software. It would just be, kind of up to the customer, right, to the user, right, to determine which one would be most appropriate for your experiment, as we've talked about, for situations where there are a lot of good high-scoring guide RNAs, maybe in those situations, then you could, opt for the wild type. But of course, in situations where there are not a lot of high-scoring guide RNA available, you can order the HiFi Cas9 through that software.
Right. Yeah. We really leave it up to the user. Yeah. So this is actually an interesting delivery question. Do I need to re-optimize my RNP delivery conditions if I switch to the HiFi Cas9?
And I assume the question is around if I switch from the wild type to the HiFi Cas9, right?
Yeah, I assume that's what I mean. Exactly. Yep.
Yeah. No, so it's the same delivery conditions for both protein, which is obviously kind of convenient, right? Because whether you're using electroporation or lipofectamine, you don't have to change the conditions. By switching from wild type to the HiFi, they both use the same delivery conditions.
Right. That's nice. And actually, it looks like our last question is kind of similar, or maybe it even came from the same person. Does it matter if some of my experiments are with regular Cas9 and others are with HiFi Cas9, or can I switch between them?
Yeah. Right. Great question. So yes, you can switch between the two. Again, because both are protein format. It's gonna depend on, like I mentioned, right? Like what are you looking to achieve? But of course, you can switch between the wild type and the HiFi. No worries. Both are the same in the same protein format. So, which is really nice, right? Like you can kind of, depending on what you're looking to address, if you have a lot of off-target concern around off targets, and you can just conveniently switch to our HiFi Cas9 protein with no impact. Yeah.
Excellent. Well, that was the last question that was submitted. Unless there's more that I just am not looking at right now, those are the ones that I captured during your talk. But so before we go, I just want to thank you one more time. If there are any questions that we didn't have time to answer live, we will address those by email using the address that was provided when you registered. So thank you again to the audience for joining us today. We also want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This will be available for viewing on demand, so you'll get a LabRoots email alert when it's available for replay. And we, of course, would encourage you to share that email with that link information with any of your colleagues who may have missed today's live event. So with that, I thank everyone again for joining us and hope you have a great day.